Hey guys, today I want to talk about some experiments that helped us understand the role of DNA in heredity. And we figured out, based on some experiments by Thomas Hunt Morgan with sex-linked traits, that chromosomes were probably carrying the genetic information. But there are proteins and there is DNA involved with chromosomes, and so which one of them is important? First, Frederick Griffith did some experiments that involved two types of bacteria injected into mice, and one type of bacteria was harmful, right, and one type of bacteria was not harmful. And as you can imagine, if you put the harmful bacteria into a mouse, the mouse dies. If you put the neutral bacteria into another mouse, the mouse does not die. Now, if you take the harmful bacteria, the S cells, and you heat kill them, right? So the cells are still there, but they're dead, and you put them into a mouse, the mouse remains healthy. But if you put that heat-killed harmful bacteria and living neutral bacteria into a mouse, the mouse dies. Somehow, it seems that this neutral bacteria has picked up information for being harmful from the dead bacteria. And it was hypothesized later by Avery and McCarty-McLeod that the transforming substance was DNA. And they based this on some other work that they did, but it was still relatively, I'll say the scientific world was skeptical at the time until more evidence came. And that new evidence came in the form of the Hershey-Chase experiment. So Alfred Hershey and Martha Chase performed experiments together using bacteria and some viruses that infect bacteria, which are called bacteriophages. Now, bacteriophages have two main components. They have a DNA component on the inside, and then they have a protein component that wraps up that DNA. And you can sort of see that here, right? So this is this protein capsid, and then inside there would be some DNA, and you can see the DNA being injected into the cell here. At this point, they didn't know which part of the bacteriophage was being injected into the cell, so they did some experiments. They got some radioactive sulfur and grew viruses in this radioactive sulfur, and sulfur tends to be part of proteins, amino acids, right? And then they grew some other viruses with radioactive phosphorus, which tends to be part of DNA, those phosphates in the phosphate sugar backbone. And they took these viruses and they allowed them to be injected into some bacteria, some E. coli, and then they looked to see if the bacteria were infected with radioactive substances. And it turned out that the radioactive substances were only available inside the cell when it was the phosphorus, right, meaning the DNA, that was labeled. And so this was some important evidence that viruses, which take over your body by giving you bad information, essentially they take over your cells by giving your cells bad information to copy, the information they were giving your cells was in the form of DNA. More information on the structure and therefore the role of DNA came from Erwin Chargaff, who noticed that the composition of DNA, meaning the levels that you have of A's and T's and G's and C's, vary between species, right? So a mouse will tend to have fairly consistent A's and T's and C's and G's, right? A human will have fairly consistent A's and T's and C's and G's, but what you find in a mouse is different from what you find in a human. And there were really two main findings. One is this variation between species, but within species, right, not only, do different, individuals of the same species tend to have the same amounts of each of those bases, but the A's and the T's are essentially equivalent, and the G's and the C's are essentially equivalent. And he did not understand why. It wasn't until Watson and Crick that we figured out the answer to that. Now, Watson and Crick are very well known for discovering the structure of DNA, so that double helix that we're all familiar with, that's on, science-y logos all over the place. But they didn't do it alone. In fact, they relied very heavily on information from Rosalind Franklin, who worked in a lab with Maurice Wilkins, and she was an expert at something called X-ray crystallography. What X-ray crystallography is essentially you make a crystal out of a molecule, and DNA is a salt. It's charged, so you can do that. You can make it a crystal, and then you shoot X-rays through it, and then they go and they splatter behind your molecule, behind your crystal, and you can get images. And this is a very, very famous image taken by Rosalind Franklin that helped show the shape of DNA, right? It was this, amongst other data, like Chargaff, for instance, that indicated to Watson and Crick that what we had was helical, right, in DNA, and that it was probably a double helix. So based on that photograph from Rosalind Franklin, they got some very basic information about the DNA. They were able to take measurements and figure out what the width was, and they realized that it was a consistent width, in addition to the fact that it was helical, which was apparent from that photograph. And so they had initially thought that you might put purines with purines. Purines have 2 rings. Pyrimidines have 1 ring. But if you do that, you get a very wide molecule. If you put 2 pyrimidines together, you get a really narrow molecule. But if you put 1 purine and 1 pyrimidine together, you get a width that is consistent with that X-ray data. And so they realized that you have to put a pyrimidine with a purine. And not only that, knowing Chargaff's data that the amount of A is equal to the amount of T, and the amount of G is equal to the amount of C in each organism, and they then paired up these sets of nucleotides, A with T and C with G, and they realized that you have some very nice hydrogen bonding, 2 when you pair A and T, and 3 when you pair C and G. That difference in number of hydrogen bonds will end up being important. And they cracked that code, so to speak. And understanding that A's always bind with T's, and that C's always bind with G's, that was a huge leap, because it indicated how these things copy themselves. Because if you have 2 strands attached to one another, and they figured out that it was a double helix, if you separate those strands, if you look at the letters on one side, you then will immediately know what the letters were on the other side, right? If I see A, C, C, G on this side, I know that it's T, G, G, T, G, G, C on the other side, sorry. And so, similarly, if DNA separates in a cell, and you add new nucleotides to each old strand, well, those new nucleotides are going to have to be the same as the old paired nucleotide, right? And so you get an exact copy of the original double helical molecule that you had, and DNA is incredibly good at copying itself with fidelity, with accuracy, better than any other molecule that we know about, and that is why it's the information-carrying molecule, right? And they cracked that case. So how does DNA replicate? Well, you start with a double stranded molecule, and it separates, right? These 2 separate, and you have to do that somehow. Now, we know that in PCR, we do that with heat. In your actual cells, we do that with an enzyme called helicase. And then you add new nucleotides to each side. And again, we know that if we have ACTAG over here, we have to add TGA-TC over here, just like the old strand. And if we have TGA-TC over here, we have to add ACTAG over here. And so you get 2 copies that are identical to that original copy. That said, each new copy has 1 old strand and 1 new strand. And so we call this semi-conservative, not because it's sort of halfway Republican, but because we've conserved 1 strand of the original one, and then put in a new strand here, and the other one has 1 old strand conserved, and then 1 new strand. So it's not entirely conserved, where you'd have 2 old strands, but it's semi-conserved. And there really were 3 possibilities for copying. Watson and Crick, based on the structure of DNA, had indicated this semi-conservative model, where you end up, each new strand has 1 old and 1 new strand, right? And then if you copy again, you have one old and one new here, one old and one new here, but these are both going to be new as compared to that first generation strand. Conservative means you would just create a whole new double-stranded copy, and this one would be conserved, right? It would have both the old strands here. Dispersive would mean you would break the whole thing up and all of these would be mixed, right? Mixed old and new. Well, which is it? Well, Meselson and Stahl did an interesting experiment later on with weights, right? And they essentially took heavy isotope and a lighter isotope. So the dark blue is heavy, the light blue is light. And they said in a conservative model, you copy it, you would expect to have one really heavy strand like the original, right? If you have a heavy original and you place it with light nucleotides, right? So all new strands have to be light. You'd expect a really light new one and a really heavy old one. And that's not what you get, right? In a semi-conservative model, you would expect something in between because you've got one heavy strand that's original and one light strand that's new in each of these, right? You would also expect that from the dispersive model, right? So when they found that these new strands are somewhere in the middle in terms of weight, that rules out the conservative model, but not the dispersive model, right? But then you copy these guys and make a third generation. And in the semi-conservative model, you would expect, essentially, when these separate, we have two old strands to work with that will each get a new strand, but these two new strands will each get new strands as well, and so you end up with two that are in the middle and two lighter ones after 2 rounds of copying. And that's what you find. In dispersive model, you would still, after that second round of copying, expect to see 3 that are in the middle. This is not what you see. So this was some empirical evidence for the semi-conservative model of replication, where you have an original strand that you break apart, and then the new strands, the new double strands, each have one old strand and then one new strand that fills it up. So how do we do this in your cell? Well, let's start looking at a prokaryote. So a prokaryote, which does not have a nucleus, right, still does have DNA in the center of the cell, in the nucleoid, and that DNA is a ring, right? You have a ring of DNA. So essentially what happens is you separate the strands right here at what we call the origin of replication, right? So these blue strands that were together, double-stranded molecule, have now been separated. And we begin adding new nucleotides to each of those strands here. And as you can tell, that creates this little bubble here. We call this the replication bubble. And on either end, you have a little fork, right? This is called the replication fork. So this whole thing is a bubble, and this bubble is getting bigger as we continue to add nucleotides in each direction. And you have a fork on this side and a fork on this side. And that bubble continues to get bigger as that fork spreads around, right? And that fork will eventually, these two forks will meet each other on the other side, and then that one strand became 2 new strands, each with an old strand of DNA, each with a new strand of DNA, semi-conservative. Now it's different in us because we have linear chromosomes, right? And ours are also much bigger. Bacterial chromosomes are very small compared to our chromosomes. And so we actually have multiple origins of replication, which means we get multiple bubbles, replication bubbles. And each replication bubble means 2 replication forks that are going to move in the direction of one another. They're going to expand and expand until we end up with 2 completely new strands. How do we do that? We do that with something called, we unwind this DNA with something called helicase. And then we have some other enzymes that we'll talk about in the next video that will help add these new nucleotides together. So to tease the next video, right, we're going to have helicase, which unwinds this double-stranded molecule. We're going to have primase, which adds primers, which are going to be important to start these off. We have single-strand binding proteins, which sort of maintain this, maintain these separate, make sure they don't kind of fall back together. We've got something called topoisomerase, which prevents like overwinding. And then we will have something called DNA polymerase, which will be very important.